All right. That always makes me feel good when your live pops up.
Perfect.
Sometimes it doesn't, and then you're in kind of a cluster. So, morning, everyone. Lance Tarchione here with you on Ask the Agronomist. Joined by my friend and colleague, Ryan Tichich. I'm going to let Ryan introduce himself here in a bit and what he does. We're coming to you live from what's known as D Building in St. Louis on campus. So, this is the old Legacy Monsanto headquarters here, now the Bayer North America headquarters here in St. Louis. Chris Cowlaw with us here, managing production here this morning, and Chris and I are in town for a big meeting for we Bear folks, and you folks out there, customers will hear more about this meeting that we've been in here for the last 2 or 3 days, coming up here over the next week or two. But pretty exciting stuff that Chris and I are learning about, and we think you'll we think you'll like what you hear coming out of the meetings here this week. So. Good stuff. That's not what Ryan and I are going to talk about. We're going to talk about other good stuff that we got going on at Bear. And as always, please ask your questions. Chris is manning the chat, and we will give priority to your questions. But with that, I'm going to kick it over to Ryan here. Ryan and I have worked together on some stuff. We know each other a little bit. We don't go way back necessarily. And I've had guests on before that I do, have 20, 25-year histories with. Ryan's actually on here because he lost a bet. He got coerced by a couple of people who've been guests on here before to help me out with this. And very glad that he's here. He's intimately involved in some things that we're really excited about, really interested in, and they relate to corn as well, which is everybody's favorite crop. Let's be honest. and so I'll quit talking here and let Ryan introduce himself.
Yeah. Hey, good morning, everyone. Ryan Tichich. I lead our corn agronomic systems team within market development, and so really what we do is we work kind of nationally with all the teams that are working on corn and really help identify what the market challenges are and think about how we bring some solutions and how can we look at the portfolio that we have and what's in the market and ultimately try to tailor some solutions, create some solutions for our growers. So often that involves like product launches, trying to bring together CP and germplasm, right, along with digital, just trying to bring that total acre solution into place.
So how you all would connect to what Ryan just said was he mentioned the phrase market challenges. That would be the things you complain to Cowlaw and I about. It's like, this stuff really sucks. Why don't you guys do something about that? So that's why he's here.
Yeah.
He's working to do something about that stuff. So we're going to talk about crown rot. We're going to talk about some of the agronomic challenges that are out there and what Bayer's doing to try to address those. I know I was at one of our research facilities this summer with Ryan, where we were looking at some new methodology we've developed to screen hybrids for tar spot. Yep. So our good colleague and friend Jim Donnelly, who's been a guest on here before, he was credited with developing something that we internally call the Donnelly method.
That's its name, right?
That's right. That's right.
Become that.
I'm not sure. He won a couple of awards. I'm not sure he's gotten any money for the Donnelly method, but he has been recognized as he deserved to be for his ingenuity of coming up with that. So anyway, you just got back from a big field trip to Arizona.
We did
with a bunch of our groundbreaker growers. So. Talk a little bit about that, what that was about, and what that facility is. And I've never gotten there yet myself. I'm not a big fan of flying, honestly. So I don't know if I'll ever make it to Marana or not, but you can tell us about it.
Yeah, I'll tell you a little bit about it. I mean, it was an opportunity to get all of our groundbreakers growers together down in Arizona. And you might be thinking, why Arizona, right?
I asked that question.
It's like, well, it is a little warmer down there. So that was a plus, right? And we look at the way the weather was over the last couple of weeks. And
well, it's good sunlight down there too.
And that really was a driver. So we've got a big greenhouse down there.
You say big, it's like acres, right? 7 acres under glass. That's pretty cool. I've got some real fields that are smaller than that.
Yeah. And so it's really the place where we go ahead and build all of our new inbreds and our new hybrids, right, that we're going to be selling. We add our trait components to all of those hybrids there. And like you said, I mean, you look at the light conditions in Arizona, and they really are optimal for crop production. and we've got access to plenty of water, and so you can, in a greenhouse setting, grow corn very reliably there. And so, yeah, so it was a great opportunity to take the Preceon growers down there. Really, just thank them for their experience here this year. I mean, they are partners in what we're doing with Preceon. It is very much a test and learn approach, right? We're kind of co-creating, and the feedback that they all provided is massively important as we shape and build the products out. So just a way to kind of say thank you and show them a little bit about how we build the hybrids and the products that they were planting in their fields. And yeah, just a good opportunity to take a look at how performance was across the board and review that with them, talk about some of the yield contest winners and things like that. And yeah, so it was really, really great.
Yeah, sometimes growers get a little frustrated with us because they think we're doing a little too much testing on their farm. We'll bring out something new, and maybe we have a few surprises. Maybe it doesn't quite perform up to what we thought it would, and like, well, you guys should have looked at that a little more before you brought it to us. Well, if you sign on to be a groundbreaker grower, that's the intent. That's what we're doing. Yeah. We're going to tell you right up front that you're going to help us evaluate this product, and we were looking for that feedback from those groundbreaker growers of, what's it like to harvest corn with an ear that's only 20 inches off the ground. What's it feel like to drive through that stuff in a high boy sprayer? One interesting observation I talked to a grower that had was typically in regular corn in a high boy, it's barely getting over the top of it. Sometimes it's bending it over to get through the field. Yeah. So you're always looking up out the glass. And so there's a whole bunch of monitors and equipment that's mounted at the bottom of the glass in the cab.
Yeah.
Well, when you're in corn that's only 5 feet tall, you're looking down, and your visibility is not very good down because the people that built the machine assumed you'd be looking up, not looking down.
Yeah.
So just little stuff like that you wouldn't really think about until you've done it is, kind of an example of, some of the learnings that we've had. And if everybody that's running a high boy sprayer starts running it through short corn, you might have to move some of the stuff around in the cab so you've got better visibility out the bottom of the glass instead of the top. So you mentioned something that kind of triggered with me. You're talking about building inbreds.
Yeah.
And so I think that kind of ties into precision breeding, which I wanted you to talk about a little bit. So, back in the old days, you might have said we screened inbreds, or we looked for inbreds, or so talk about how building one is different than looking for one.
Great question and lead in. Yeah. I'll ask you a question maybe. How many genes are there in corn?
Oh, good grief. I don't even know.
Is it more or less than what a human would have?
More.
It's
right?
more.
Yeah.
That's right.
Yeah. Yeah. I don't know the number, but I know it's, we think we're complex, but corn is more complex.
Exactly.
I guess.
I think humans have something like 20,000 genes and corn has 32,000.
Yeah.
So if you think about 32,000 genes and then the number of genetic combinations that you can get from that, it is a massive number. I can't even articulate the number with words, but I'll just tell you what it is. 200 with 21 zeros behind it.
Oh, wow. Okay.
Right? That would be the number of genetic combinations that we can make in corn, right? When you think about those 32,000 genes, and then you think about how many environments are out there. Well, that's a pretty big number too. It's 3 with 15 zeros. And to me, that's what, precision breeding is all about. Like you said, we're not just throwing a bunch of inbreds out there, testing them in different places and then seeing what kind of comes to the top. It's learning more and more about, which genetic combinations are the right ones for that particular environment that we're trying to address. And then saying, okay, how are we going to get those genes? And let's go ahead and actually build the inbred that contains those genes, which then becomes part of the hybrid and ultimately leads to success. So I think in the past, we looked at it like we're advancing hybrids through, or we're advancing inbreds, right, as part of our process. And now I think it really is about we're pushing genes, right? We're building genes and building inbreds that are going to be successful in environments X, Y, or Z, right? Depending on where we're at. So it's that conscious decision to build and design an inbred that's going to meet the need of that particular grower.
Right. Well, and we try to get we get feedback from our customers all the time. We were talking about that earlier. You know what you would like to grow. Sometimes, you really like this hybrid, except it has poor emergence, or except it's really susceptible to green snap. We have the ability with some of the technology that's emerging to maybe genetically edit, products. And I had a really fascinating conversation with a guy that was an ag tech presenter a couple of years ago talking about gene editing. And when you look at the throughput of the breeding program today, I kind of wonder, well, gosh, with that much throughput, even with all that genetic diversity, are we someday going to run out of genetic diversity? And we've checked everything that can be checked. And he said, there's a lot of genes that have been silenced in the corn genome that we might be able to turn them back on,
unlock them,
and create diversity even in the genome. So he wasn't worried at all about running out of genetic diversity with the technology that exists.
There's so much out there. And then I think, again, so that's one part of precision breeding, right? Is how do you design those inbreds with all the combinations that are out there? But then I think the other part is really the data, right? And we just have so much more data today than what we would have had 10 years ago, 15 years ago. And I think it comes through different forms. Some of it comes from, customer feedback directly, right? They're telling you and Cal-Al, and we're taking that back to our pipeline. Some of it comes through things like,
and when you chew on us, we chew on somebody.
Right.
So it gets passed
the
along.
feedback comes, it sure does. And it gets taken into account. Absolutely. But field view, right? And we're more and more digital, and we can talk a little more about groundbreakers and some of the learnings that we've had by region and about some of the hybrids. That's information that we're able to take back, and it really is about mapping. Okay, that hybrid had these genetic combinations in these environments. Here was the result, right? And you just repeat that time and time again across all of the places that we test, all the hybrids that we have. And that digital flow of data is something that's new. As we field test, right? We have a huge field testing network in our breeding program. They are collecting more and more data from their locations, right? That gets mapped back to the genetics. And then once you kind of establish the relationships of what genes are successful in which environments, you can start to extrapolate that back to some of your previous data, right? Before you know it, you're generating a huge model, if you will, on how to stack these things together. And then there's the whole simulation front, which is maybe new, right? Where we're modeling out simulated environments, things like that. We call it digital twin that just supplements all the other data that we have. And so you've got all that data flowing together to help make a better decision on which genes and genetic combinations to promote.
I guess I would describe in layman's terms that, digital twin is, you're essentially, you're virtually testing hybrids.
Correct.
And trying to predict, okay, we think this would be awesome.
Yep.
We should build that.
Yep. And some of that is based upon, hey, at closely related locations, X or Y, we saw these hybrids do this. Well, if we tweak that and simulate a slightly different environment over here, maybe with different water, different things like that. Okay, what additional information do we learn that we can then apply to the decision that we need to make?
Yeah. One thing Cal and I heard about it at Ag Tech here a couple weeks ago was, we've got a program coming that's going to allow us to sort through all of our data connected to weather.
Yeah.
Which has always been a big miss, in my opinion.
Yeah.
It's fairly easy to get weather data on every square foot that's out there.
Yep.
But we haven't had a good way to tie it back to, okay, what yield did that weather produce?
Exactly.
And, looking at things like temperature, nighttime temperature, rainfall, those are the biggies. People ask questions about sunlight and other things, but typically corn yield is going to relate to moisture and temperature.
Yeah.
As far as the weather goes.
And we like to, and we look at soil types and things like that. And those things are absolutely important, but sometimes differences in soil type depend on the weather, right?
Right,
Certain
right.
years you get rain, that soil type
Right.
difference may not matter.
Right.
Other years it may matter quite a bit. So I
Yeah.
agree with you that
Yeah.
that layer is tremendously important. And that's the kind of data that we're gathering more and more, not only from commercial fields, but like from our breeding sites and things like that. And so I think it's a lot more data, and that's just all on the characterizing the environment. Of course, genetically, right, through the genotyping that we do and things like that. There's a lot more data on the genetic side, and you just how we're stacking those two things together to, again, think about how we want to design that inbred is really, really an important piece. But the other part of it is then once you make a decision that I need to build this inbred and match it to these environments, how do you build that inbred as quickly as you can? Right. And that's where Marana, the greenhouse, comes into play, really, right? Because you need to minimize, we can do 3 cycles a year, right? And so we can start to, as soon as that decision is made and that combination is unlocked, we immediately put it into action at Marana and start that process of actually building the inbred and then getting it ready to ultimately, eventually get traits and get out to a grower's field. So we can do that a lot faster than we could have in a traditional model where it's, hey, you make the cross, then you go through several cycles of selfing it down, right? And then eventually we're going to field test it. We got to do that for 6 or 5 years, whatever. And then eventually you get a product at the end. So we say we're 15 times faster with kind of this precision breeding process that, again, Marana is a key tool and component that bolts on to the whole concept of how we're kind of building the combination.
And I think sometimes when, and maybe I'm projecting the way I think on farmers, but, sometimes when I hear faster, I get a little nervous that, well, are you
sure?
Going so damn fast, you don't know what you're doing, right? But the positive side of being able to do stuff fast is, we've got emerging problems that need to be fixed. So, Goss's wilt was one, a few years ago. Tar spot is one now. Crown rot is one now. And, in the old days, it would have taken you maybe 10 or 15 years to genetically fix a problem like that. But we can probably do it in a fraction of the time today. So as it gets harder and harder to raise 300 bushel corn because of some of the curveballs that Mother Nature throws at us, we should be able to address those faster with this technology.
I agree. And you're absolutely right that the testing piece remains critical, right? I like to think of it as like the front end of the pipeline, right? We're using the data, the analytics, all the things we were talking about to go faster, right? But there's still that end point where, hey, once we get the traits, we need to follow through with, multiple years of solid testing. Obviously, the agronomy team, right, ends up kind of throwing it through the hurdles as well, right? And so those are still critical pieces to make sure that we get the products perform the way our customers need them to. But agree with you, it'll be an outstanding tool to, hey, when new challenges emerge, right, we can be more agile than ever in addressing them.
Good stuff. So I guess other things that are important to growers that you and I have worked together on. I know you've done a lot with seed quality in the past few years, and we've done a lot of testing to try to make our seed quality as robust as we can. And there's a trade-off there that I'm not sure that everybody understands. That if you keep the best 10% of the seed that you produce, it's going to be awfully damn expensive. Some people say it already is awfully damn expensive. And if you keep too much of the seed that you produce, that means you're selling some stuff that's not quite up to snuff. And where you draw that line is a really difficult decision to make for the customer and for us as a business. And having good systems and metrics and tools and procedures and tests and stuff to know where to draw that line. And you maybe don't even draw the line in the same place on every hybrid, because hybrids are different, and vigor and genetic differences in hybrids. I've seen this many times. Poor quality seed of a strong emerging hybrid looks better than high quality seed of a weak emerging hybrid.
Yeah.
And so, there's so many factors that go into that. And, we're working to develop some models to try to help people, maybe make better decisions around planting, which that's really tough for a grower to do, because if the model says you shouldn't plant today, but your neighbor is, you're probably not going to pay attention to the model. And having the discipline and the patience to maybe leave the planter in the shed on a day when, hey, based on soil temperatures, based on calendar date, based on the upcoming weather forecast, based on soil moisture, based on this, based on that, your odds of having a great outcome are pretty low. And you don't necessarily have to not plant because of that. But we'd like to make you aware of the fact that, okay, this is probably a risky window to be planting in. If you want to, you're a big boy, you can do what you want, but maybe we ought to think about that.
Yeah. I mean, and everyone, I think, realizes, right? I mean, the planting windows seem like they're moving up. Equipment's getting bigger, right? Acres go into the ground fast. And so when conditions are good, people put a lot of acres in quick. And I think some of the things you're talking about, we're saying we don't want people to plant early. Well, not necessarily. We realize that's part of the business, but can there be some awareness as to what the risk is, right? How far do you go, April 10th versus, April 20th, when you look at kind of the risk of what the next 10 days look like. And I think some of these models just help agronomists, help growers, help everybody think about, okay, here's maybe an appropriate level of risk to take. And maybe that, as long as we're getting people to think about that relative maybe to the hybrid, like you said, some are stronger out of the ground, some are a little slower out of the ground. All those things kind of come into play to make the right decision there. But it's been a fun project. It's been a great project. It's one that, if you think about it, really spans across the whole company.
Right.
Because I mean, you've got product supply, right? They're building the hybrids, and we've got breeding, developing the hybrids, and so those two things have to work together. How we, the agronomy team, right? How you guys work with it, FSRs.
It's been a little bit fun, and this is I'll share a little bit of dirty laundry here. It's a little bit fun to watch the various groups, So the manufacturing folks that are producing really high quality product might think that, well, breeding, you need better you need better emergence. And, breeding might think, well, emergence is fine. You guys need to take better care of the seed. And maybe the seed treatment guy's like, well, I think I can fix it all with a better seed treatment. And so everybody's kind of got their part in that system. They do.
There. And I think the cool thing over the last few years, right, and it is really just trying to say, hey, let's put the customer at the middle of all that, right? Yeah. And now you've got the breed, everybody's kind of looking at, okay, how do we do our part to maximize the experience for the grower, create the best experience possible.
Cal and I and the grower don't care why it came up better than Pioneer. They just want it to, So it could be the seed treatment, it could be the germ, it could be the quality, just get it out of the ground.
No, but we've done a lot. I mean, so you mentioned seed treatments, right? We'll talk about that a little bit. I mean, so EDC is something now, our enhanced disease control platform is on a great many of our products at this point, all of DeKalb.
And Chris and I were early promoters of EDC, and we're very happy when we just decided to put it on everything, because no matter how much Chris and I talked about it, and no matter how much good data we shared with people, nobody really wanted to pay more for it. And so we never got it on much seed until we just made it kind of
the standard. And it just improves early season performance, and we have a lot of great data saying that it helps with emergence and vigor, which is a win. We added, of course, ethoboxam, which is a fungicide that helps out with
which that makes 4, right?
It makes 4
different fungicides on Acceleron. So just trying to get the best seed treatment we can on that seed. I mean, there's a limit to how much stuff you can throw on a seed.
There is.
And we're probably getting
so much space.
we're
only so much space.
We're probably getting close to that with the treatment that we've got on our VT4 Pros and Smart Stacks Pros, which we'll talk about that at some point as well. But a lot of work goes into seed treatments, and going back to the seed quality thing. I mean, you can't you know, if you got poor quality seed, you really can't fix that with the treatment.
Yeah.
You know, the treatment helps, but you got to have the good quality. Yeah, it is interesting to analyze, and we've looked at this where you have complaints, where you have replants, where you have problems with emergence, correlate that back to what was the seed quality of that lot of seed, and typically there really isn't much of a correlation there. We have the best quality seed we have that fails due to environmental conditions, and we have situations where seed that we knew when we sold it was barely good enough to sell is perfect because of the environmental conditions. And so the environment that seed goes through after planting actually trumps how good the seed was when you planted it. But you know, that's still important, and we're still trying to figure out what's the best way to manage that. And every farmer out there wishes they could pick the best quality stuff. And we get a lot of pushback from growers about, well, why aren't you guys more transparent about what your cold test scores are? And we've had that conversation on Ask the Agronomist before. You know, some people go to the trouble to send seed off to third-party labs to get tests done. We've looked at that. You know, a lot of that stuff is it makes people feel better. It gives people information, but a lot of times the wrong decisions get made off of that information, because the weather's really what's driving, whether you get a stand or not, more than the hybrid or the seed.
Yep. No, absolutely. And I mean, just tipping the hat to the agronomists out there, right? I mean, you guys have helped us characterize some of these complaints, some of these replants and things like that. Because there's even, you need to have really good data, right? About where you saw the challenge, what the challenges were, right? And we've done that over the last few years, acquired additional information about where we've seen some challenges. And that, I think, in turn has fed our proprietary cold test process, right? We've been able to refine that as part of some of the changes we've made here over the last few years. And as we validated that, right, because we'll take different quality levels and plant them into very tough conditions. And I think we've shown that this test does a really good job of discriminating there between good and poor, and that is something that we've definitely built upon in our process for deciding, hey, are these things good for the market? Are there any watch outs, things like that we need to be thinking about there? So so I think the other things we've done is we've looked at the higher risk maybe areas, RMs and things like that, and just as we've looked at that proprietary cold test, if we've had some things that are on the fence or closer, right, there's been a lot more internal review of because we'll pull in representatives from agronomy and also product management, right? Everybody kind of gets around the table and says, okay, like, should this go, should it not? Right. And been a lot more intentional in that, I would say, in certain areas here over the past few years, which all of that helps.
And it's a really big challenge for any seed company too, because the tighter you turn the screw on quality, the smaller your supply pile gets. And when we're sold out of the hybrid that you wanted, you wish we could make more of that. And if you lower your quality standards, all of a sudden there's more. And so there's that tug and give and take on supply versus quality, and you can't sacrifice quality to create more supply. But a lot of times, I've had growers that have told me, hey, if you've got something that the germ's not quite as good, but it's the right hybrid, knock $5 off of it and sell it to me. And we've actually done that in a few, rare circumstances, but it's not a habit that any seed company wants to get into. And it's, like I say, it doesn't. You reference those different those quality trials that Chris and I have been involved in, and the way those are set up, we get 4 different quality levels of different hybrids, A, B, C, D, and A is obviously the primo stuff, B is really good, C's maybe close to the threshold
Yeah.
of quality, and D is, we wouldn't have sold it.
Would not be approved. Yeah.
And it's honestly been surprising to me how often there's no difference in emergence between those four quality.
Yeah.
And there's big differences. If you sent those 4 lots off to a third party lab, a couple of those would make you go, oh, you know what, that's, I don't want to plant that stuff. And it's fine. Unless you get
the wrong
conditions,
Yeah.
And there's been plenty of times where I've taken the stand counts on them, and B or C quality may have a better stand.
Yes,
than A.
I have too. Yeah, same thing.
And you
Yeah.
scratch your head. How did that work?
Yeah. I mean, I always rate them blind. I don't know what I'm in A, B, C, or D when I'm rating them. And you take and you're seeing some differences in stand counts. And I count runts too.
Yeah.
That's another thing I do, because I think sometimes quality, you might have the same stand, but do you have the same uniformity?
Well, uniformity is so critical.
And
So,
guys get really, really, really upset about non-uniform corn because we make a big deal out of it's good for it to be uniform.
Yeah.
And so I'll take these ratings and I'll think, oh, there's some differences here, and I'll match it up with the treatments. Like, well, there's differences here, but they don't make no sense.
Right.
Yeah.
Right. I mean, and it's like we were just talking about, right? I mean, yes, the quality is an important part of it for sure, but then the genetics too, right? Was there something about the genetics in that environment that just led to, even though that lot was rated a C, it just did really well there.
Yeah. Well, and we see that with, it's always been funny to Chris and I, when like new hybrids every year, we come out with agronomic ratings on the new hybrids. And then after that first year, we tweak them sometimes because we learn things.
Get more experience.
And we've been on calls with agronomists all over the country. And some guy in Nebraska is bragging about how awesome the emergence was on this hybrid. And Chris and I thought it was awful. And what's up with that? Well, it's a lot wetter in Illinois than it is in Nebraska. And I think a lot of times how plants handle excess moisture is more important to Chris and I than temperature. What ruins our stands is too damn wet.
Wet feet.
It's not too cold. We're in Illinois. Hell, we raise corn in Minnesota, North Dakota, and Canada. I guarantee you, there's places colder than Illinois. So it's not the temperature as often as it is the moisture. We just get too damn wet, and you can have the highest quality seed in the world, and you can still suffocate it. A corn kernel can't hold its breath very long, and that's what causes us trouble, probably more than quality or temperature. And not a lot we can do about that. So, well, while we're on the, I should call out too. So 7:32 now. If you're just joining, because we normally start at 7:30, and you're wondering how long they've been going here. Well, we've been going 30 minutes. We started a half an hour early today to accommodate our guest schedule and to get Cal and I back in our meetings on time. We were told that it might be bad for us if we were late for the meeting. So we need to be on time. So if you missed the first 30 minutes, you can go back and watch the recording later. Welcome to those of you that are maybe just joining, and please, if you've got questions, chat them in. Chris will get them on the chat. So we were talking about seed treatments. We haven't talked about N314 yet. One of our more brilliant names for a product. Right? So tell us what N314 is.
It's so I think maybe people are familiar with Poncho Votivo, right? And the Votivo part of that is a biological nematicide. So Bacillus firmus species, and so that has been our flagship nematicide product in the past. Well, as we move to N314, what N314 is it's the vote.
I said it wrong. It's 314. It's not 314.
Both are catchy. Both work.
And I always joke that somebody from St. Louis come up with the 314 because that used to be the original area code.
St. Louis. There might have been a vigorous debate between 636 and 314. 314. I don't know. Our marketing guys, So I'm just kidding. No, but
I've never had one of them as a guest on here.
Yeah, maybe that'd be interesting to hear their insights, right? How did you guys decide this? No, it's all the Acceleron kind of components have
Mhm.
those
Mhm.
rather nondescript
Right.
names,
Right.
right? So anyway, we're not usually intending to market the components. It's more about the Acceleron name that we just kind of bring the best components together under that brand. But no, N314, it's got the Votivo part of it. So we have that biological nematicide piece. There's also data to suggest that really helps us with some root growth development, things like that, more root hairs, right? All that is part of what we were just talking about. Try to really get a good stand early, good vigor early. Votivo is an important part of that. So 314 has Votivo, plus it has fluopyram.
Mhm.
Okay, and fluopyram is a chemical nematicide with a pretty good track record. We use it quite a bit in soybeans under the name Ilevo. And it also has some fun.
Now, I thought that was a fungicide.
Yeah, exactly.
Yeah.
I know, right? So it gets a little confusing, a little tricky, right? Fluopyram is just an interesting molecule from that standpoint. It's a nematicide and it's a fungicide.
Well, and the story that I heard was, you can correct me if this is wrong, I mean, that's a legacy Bayer product.
It is.
And when they were developing that, long before the three of us had anything to do with Bayer, they were developing it as a nematicide.
Yep.
And accidentally discovered that it's also a fungicide.
Yeah.
So even though it's probably more known in the market today as a fungicide, it was first developed as a nematicide.
Well, it's an important part of like Delaro Complete, for example, right? So I mean, it's a remarkable compound that way.
Matter of fact, I think it's in so many things we have a hard time making enough of it. Right?
Yeah, we do. I kind of joke about it sometimes that it's the aspirin of like crop protection because it's in fungicides. I mean, we use it in a lot of different places in furrow.
Well, we actually keep discovering new ways to use it.
New ways to use it. Exactly right. So N314 brings that fluopyram piece. It has the Votivo. It really is the only product in the market from a nematicide standpoint that's fully systemic, and moves within the plant because of the fluopyram component. So the history on it, I can share a little bit of those details, right? I mean, we're searching for that next gen nematicide, and we wanted to use the fluopyram as a little bit of a control in corn, and turns out we saw really, really good results with it. So we just worked working with our partners, BSF, to figured out a path to be able to bring that product in, and very happy to be launching that and getting it out broadly into corn. Obviously, we expect good performance from a nematode control perspective, and again, of course, are places with very severe infestations, and sometimes you've got to do more than just a seed treatment there. I get that, but there's a lot of acres that kind of have that subclinical, impact where the nematodes are there. They're not showing a lot of symptoms on the plant, but they're grabbing some bushels
on a lot of our soils in Illinois. I mean, good soil does a pretty good job of masking the damage that nematodes do. You get on the sandy soils, not so much. You've got heavier nematode pressure, plus you've got a less forgiving soil. And I've seen in some severe infestations, I've seen nematode damage worse than corn rootworm damage on what it'll do to a corn root system, to where corn really just can't even survive. And a seed treatment's probably not going to fix that with that kind of pressure. But and probably the first question that came to some growers' minds is when you mentioned the connection to Ilevo is 314 gonna ding my corn, gonna burn my corn, gonna stump my corn, gonna
we do have the halo effect in soil.
Yeah, that's right, and that's a
sign that it's working.
I've always thought halo effect was a cute little name for what's not all that cute sometimes in the field. So
very true.
So the phytotox that we sometimes see on soybeans,
we have never
not
seen
an issue in
never
corn.
seen it.
And I think we've even got some data.
Nor in cotton.
We've got some data. I think we've even maybe seen a little bit of a vigor enhancement sometimes.
That's correct.
And whether that's due to the chemistry or due to the nematode suppression might be hard to say. But we have not seen any detriment to the emergence, early growth, vigor in corn of the addition of that chemistry to a seed treatment. So, the way we are introducing, that product into DeKalb corn is in, our two of our newer rootworm products.
That's correct.
So Smart Stacks Pro and VT4 Pro, which a lot of you probably haven't planted VT4, maybe haven't even heard of VT4 Pro yet, and maybe haven't planted Smart Stacks Pro yet, unless you're from Northern Illinois, where you're desperate for anything that will kill a rootworm. So those two trait stacks are going to come standard with N314.
That's correct. That's correct. Then we're going to start there, and then over the next few years, you will see it branch out into additional trait packages as well. The other thing that will come with it in 2024 will be Preceon. Preceon will also have N314 on it.
I don't know that I caught that.
For this crop year?
For this crop year.
Yeah. My market is actually, looking forward to above ground only products. Because our primary nematode market is not a rootworm market.
Yep. So they're coming. I mean, so as you said, we use fluopyram in a lot of places, and we find, hey, we've got to continue to get supply of it to ramp up all the uses. The other thing I'll call out is, you're absolutely right. The two new trait packages that we're launching was a great place to start, because you don't have a lot of seed that you got to go back and treat. So as you produce that new package, you can just hit it with the N314 right out of the gate. As we navigate Tricepta, maybe moving into Double Pro, things like that, we suddenly have to figure out, how do we overtreat? How do you kind of go back?
And because it's always tough to, neither one of those is an ideal situation. Overtreating is not good. Having the same hybrid with two different treatments on them is
also
not good.
A challenge, right?
So it's when you're doing stuff that has inventory, that does complicate the treatment stuff.
It does. But excited to see where that goes. And like you said, we continue to look at what else is the product doing, right? And so knowing that fluopyram is technically also a fungicide, right? That brings us to 5, on some of these products that have N314. So, just continuing to look at, we do tend to see a little bit more vigor. We've seen a nice yield response in our trials, right? Because what we'll do is we'll start with just naked seed, basically, and we'll go ahead and add all the different components, same batch, same lot, same hybrid, all that. And over time, we've seen, 4 to 5 bushel type lift from having it, which is remarkable, right? That's 3 years where we saw that. And
if that's real, I want that on every hybrid.
Exactly. So it's doing good things. We know that, right? We see the vigor. We know it's a nematicide, and continue to kind of explore what else it could be helping us out with there.
Cool. Yeah. Good stuff. So while we're talking about fungicides, diseases, you mentioned that with N314 with fluopyram on there, that's going to get us to 5 different fungicides on seed treatment. Let's talk a little bit about crown rot.
Yeah.
So, that's obviously something that's been near and dear to Chris and I's heart in a bad way. And so, so we've looked at a lot of different things. We've looked at genetics. We've looked at foliar fungicides. We've looked at seed treatments. We've looked at in-furrow products. We've looked at a lot of stuff. And honestly, in Chris and I's experience, genetics has probably been the biggest factor. A VT foliar fungicide, surprisingly to people sometimes, has been the second biggest factor. A lot of the seed treatments and in-furrow treatments, have frankly been a little disappointing in their ability to snuff out crown rot. But we do have a lot of work going on, with some university partners. Internally, breeding, genetics, seed treatments, all hands on deck to figure out, okay, what is going to be our solution to this, what seems to be a worsening problem.
Correct. Yeah. And it really starts with understanding more about the biology of it. Right. And And I think that's the part that there are things that we know, right, and that we've had experience with and generated over time. But that you mentioned the university partnerships, and I think those are so important to help us understand more about which species are actually causing the disease. It's likely a fusarium, but there could be other species, in play there.
I'm a big believer that it's more than just fusarium.
Yeah.
And I get a little nervous that we get too focused on
just fusarium. Exactly.
I'm not a pathologist. I just pretend to be one sometimes.
Yeah, and then it's when does the infection occur, and all those things are important to understand.
Well, I understand we have learned maybe a little bit about when the infection occurs. Have you been privy to any of that? Is there anything you can share there? Because that's a question that Chris and I have often wondered. Is that part of the reason that a seed treatment has limited ability to help a lot because the infection is occurring later than we thought, or
there are some you may know.
I don't. I honestly ask. Sometimes I ask questions that I do think I know the answer to. That one I don't.
but what I would say is, I mean, we believe it happens early.
Yes, I always have as well.
Yeah, and but we could be proven wrong on that, right? And that's where I think the work with the universities will help us parse that detail out, because it's an important one. But I think the working hypothesis is you've got wet feed early, like you guys often would have, and that infection occurs and maybe stagnates a little bit for a while until conditions are right later in the season.
And I would say, generally speaking, our impression has always been that the infection does happen early on. But to Lance's point, that you kind of start to doubt yourself when you think about the volume of seed treatment that we're putting on seed, and as good of products as they are, it's sometimes kind of hard to believe that can it overcome the seed treatment and be that impactful. And so maybe is it, that's where this notion of is it happening later potentially, because as the seed treatment runs out of its half-life.
Either that, or if you think about herbicides and weeds, I mean, not every herbicide has efficacy on every weed. Maybe, the seed treatments we're using don't have efficacy on the right, there's thousands of strains of fusarium.
Even within just the fusarium species.
Right.
And prothioconazole, so that's a part of EDC, is great on a lot of fusarium species, right? Especially at the rate we use. But maybe it's that one subspecies or strain that just can get around that. I mean, it's
and sudden death in soybeans is a fusarium as well, different species. But and that's flupyram is pretty good on that species of fusarium. But if it's different species of fusarium that are leading to crown rot in corn, or if it's rhizoctonia or anthracnose or red root rot or any of the other pathogens, I mean, it's going to be tough, I think, to chemically attack that complex of diseases, because it might be hard to find chemistry that has activity on the right 17 things to really fix it.
And then, I mean, so back to the question of when, right? I mean, so what about things like nematodes, right? We were just talking about nematodes. What about things like corn rootworm feeding, right? I mean, so you create damage to the root. Is that actually creating a vehicle for different pathogens to come in? Maybe even a little bit later than what we typically think, right? If that's happening, maybe at V4, V5, something like that, maybe a little bit later than emergence and things like that. So it's complicated. The work that we're doing with UNL is great. In fact, we're going to actually set up a call with Tamara Jackson there in her lab with a lot of the agronomists and TDRs in the organization here in probably about a month's time.
Okay, cool.
And just share a lot of the results, and we'll kind of go from there. But a little bit of a teaser. I don't want to spoil it too much, but they're starting to see a little bit of a correlation with nematodes. So thank you for all the samples that you guys all provided to us.
I heard that, and my first thought was, oh my God, Davey's going to go crazy.
Yeah, exactly. I've been having to curtail his excitement a little bit on it.
Inside bear joke there, but if Davey's watching, he'll get a kick out of it.
He's probably watching. Of course
I'm sure he's watching.
Yeah,
big fan of
No, I think we're starting to learn some things like that. Dr. Allison Robertson up at Iowa State is also working pretty hard on this, and her lab is generating some new information, and we're going to try to package it all up here in a month and get it out to you guys, and ultimately out to your growers. And but if we can kind of figure out a few of these biological pieces, right, then I think we can accelerate the screening process, right? So we can get more consistent conditions to be looking at germplasm, maybe other seed treatments in furrow, right? We can, who knows, maybe rotation. I mean, there could be other cultural things we need to do agronomically. But that's the hope, is we can continue to build strength through that.
I mean, and we've talked a lot about this on Ask the Agronomist before. I mean, Cal and I have identified lots of cultural things that definitely make crown rot worse.
Yeah.
The biggest challenge is some of them are also kind of important to raising high yield corn.
Yeah.
If you fiddled around and didn't plant any corn till the 20th of May, crown rot wouldn't be a thing. And when crown rot's really bad, corn planted May 20th will beat corn planted early April by 50 bushel sometimes.
Yeah.
So, if you knew how bad it was going to be, you could, let the planter set for 3 weeks and help yourself.
Yeah.
But, that's a big risk to not plant early when you get the opportunity to plant.
It is.
High populations make it worse because it adds additional stress late. That stress during grain fill is absolutely critical to it becoming a big deal. I think every plant gets infected every year with some level of crown rot. But, there's probably circumstances that elevate the level of infection. And then the type of grain fill that you have is what causes it to really explode.
Yeah.
And that's
goes
how
from being subclinical to you've got a ghosted.
Oh crap.
Plant, right?
Yeah. And that aspect of the importance of the stress through grain fill, that's what a VT or R3 fungicide, how that might help. Just alleviating stress, keeping your factory healthy. We had a year in 21 where crown rot was really bad because southern rust and tar spot killed our leaves, and you lost photosynthetic capacity on the factory. And that added stress, and then stress took it out at the knees when the crown rot took off. Well, then in 23, the stress was more drought and more environmental stress. So you got to have some level of stress during grain fill to really make it explode on you. And, that infection's been in the roots for a long time. We don't exactly know when it gets in there, and does it keep getting in throughout the entire growing season, or is it initial early infection and then it just kind of lays there and doesn't do much damage until late in the season?
Right.
Those are the things that we're trying to learn about. I've had a lot of growers ask me, well, why does it seem like it's getting worse? And I don't, it's nothing in crown rot is new.
Yeah.
Those pathogens are, older than corn. They've been in the soil for forever. So are the pathogens that cause it getting more prevalent? Is it weather patterns that are changing? Is it farming practices that are changing? Have we, kind of just inadvertently gotten into some genetics that's more susceptible to it? Over time, don't really know the answer to that. I think one thing that makes it different is in the day, we talked about digital technology earlier. In the days before yield monitors, when the combine went through that bad spot of corn, you knew it was bad, but you weren't watching the yield monitor screen that showed you it was yielding 120 bushel less than where the corn was green. And so, we see this with field border syndrome, which we've talked about before as well, which also relates to crown rot. You pick that stuff up on field imagery, on NDVI imagery. And so, imagery, yield monitors, we just manage our crop more intensively than we used to. I think the problem's always been there, and maybe it's gotten worse, but we have the ability to quantify it, see it, and know it, and you know how bad it's hurting you. 30 years ago when the corn died and it didn't yield as well, you just thought, well, corn died and it didn't yield as well. It was probably crown rot that did that 30 years ago. But we weren't talking about it.
And now, right, we highlight we can see what the impact is.
Right.
And that helps us to sharpen our tools, right, to kind of
Mhm.
go after it, and which just maybe continues to unlock, hopefully, increased yields, right? Keep pushing those yield plateaus up. So,
Chris, you had a question come in on the
Yes,
chat.
we did. We had Ross Dumbacher was asking about, are we doing any work with RNAi technology? And is there anything in the pipeline there that is efficacy or can help us with nematodes? That it's kind of
a biotech nematode approach.
Correct. Yeah.
I think early in biotech, right, they've got a lot of research targets that kind of look broadly at different insect problems. Nematodes, I'll have to go back and take a look at that one. There's nothing imminent there from a biotech standpoint, say within the next five years.
And that would be a tough one for a couple reasons. One, nematodes are, they're an animal, not an insect. And so if you think about chemistry wise, things that kill nematodes historically have been pretty toxic stuff. I mean, all the old nematicides that used to be on the market are all no longer on the market for various reasons. And so, if you develop a trait or a chemistry that controls nematodes, you got to do a lot of work to make sure that it's not doing other stuff that you don't want it doing. And I'm more closely related to a nematode than I am to an insect. And so from a human toxicity standpoint, from an environmental, issue standpoint, nematicides just are tougher to work with than herbicides, insecticides, fungicides, other things.
Yeah.
Okay, so we are getting close to time here. Ryan's got a scoot right at 8 o'clock, and Cal and I got to get to our meeting so we don't lose our jobs. And we appreciate you joining here with us this morning. And Ryan, thank you very much for joining.
Thank you, sir.
It was a pleasure, and we wish we could do these guest things on Ask the Agronomist more, and we continue to try to find ways to make that easier to do, because generally they just have to listen to me. And that's just, you can ask my wife. That's awful. So it's always nice when we're someplace where we can get a good guest. And thanks to Ryan for joining here with us this morning. Thanks to you all for joining. We'll be back with you in 2 weeks, which I haven't looked at my calendar. So, producer, that's one of your jobs.
Producer's not on the ball. Thursday, February the 8th.
Okay, so next episode, Thursday, February 8th, we'll be back with you. Probably won't have a cool guest like Ryan, but we'll hopefully have some good content. And thanks again for joining.